Hi, today I am going to present the basics of next generation sequencing and their application in the clinical purpose. So, as you are aware that these days, since last one decade, NGS is one of the most demanding application for biological and clinical research. So, this lecture will cover the basic background about NGS and its applications, especially in clinical purpose also. So if you are familiar with this image, you can clearly see here the sample extraction, that is DNA or RNA, to fragmentation and adapter ligation, then cluster growth. But there are lots of processes which are involved in the sequencing. So please subscribe our channel to get update from time to time. There are two channels. One is basically for longer videos, which I have recently launched, that is Change by Science. And another channel which includes both long and short videos about all the tutorials which I have carried out in the past 7-8 months. So please subscribe both the channels to keep updated. And now it comes to the definition basics of NGS. Next generation sequencing is basically a high throughput DNA and RNA sequencing technology that can read millions of genetic sequences simultaneously, allowing for rapid and large scale analysis of the genomes. It is widely used in the research and clinical applications for the disease diagnosis, genetic variation analysis, personalized medicine, and therapeutics. The key steps involved in the NGS processes include DNA RNA extraction, library preparation, sequencing, and bioinformatics analysis. If you are working in the lab, then your main concern is up to DNA and RNA extraction. But the library preparation is mostly carried out by the service provider, that is NGS service provider, from where you are taking the sequencing service. Then it starts sequencing processes. And the last part is computational or bioinformatics analysis, which mostly is carried out by the laboratory member itself, but sometimes also the service providers and sequencing service providers also supports up to basic analysis of the data set. But if you want precise and minute analysis, then you need to be aware of bioinformatics analysis and approaches so that you can fulfill the major goal and achieve the goal of your design project. So the key steps, as you can see here in the figure also, that is sample preparation and pre-processing, library preparation, sequencing, and bioinformatics analysis, or you can say analysis part. So next part is how it works. It is basically massively parallel library preparation, then sequencing and data analysis. What does it mean with the massively parallel? It means that unlike all older methods, that sequencing that sequence one fragment at a time. NGS sequences millions of fragments at once, drastically increasing the speed and scale of analysis. In library preparation, DNA or RNA is first fragmented into smaller pieces. Adapters are attached to the ends of the fragments, which are necessary for sequencing and include an index to identify the sample. Sequencing. The prepared fragments are sequenced in parallel. The specific method varies, but it generally involves using fluorescently tagged nucleotides to determine the order of the bases in each fragment. Now it comes to the data analysis. So before you enter into analysis, you need to remove all the adapters which you have added here. in the during the library preparations, then it is really good analysis. So sophisticated bioinformatics tools are also available, but you can also work on your own approach and design modules to assemble the millions of short sequence reads into a complete genome sequence, and to identify the variations or mutations or differential gene expression profilings, or also the epigenetics, for example, transcription factor binding site enrichment, or the methylation profiling. Now it comes to the application advantages and applications. So it helps in speeding up and the scaling. NGS can sequence entire genomes in a relatively short amount of time, providing comprehensive data that was not possible with older technologies. Now, if you want to analyze something more, it is also the cost effective. The cost of generating sequencing data has dropped significantly with the advancement in the technologies, making large scale genomic studies more feasible in terms of economy. Clinical application: NGS is used for disease diagnosis, predicting disease risk, and informing treatment decisions, particularly in oncology and rare genetic diseases. Research applications: it is a powerful tool for studying a wide range of biological systems, from cancer and infectious diseases to plant and animal research. Sensitivity: the high sensitivity of NGS allows for the detection of mosaic mutations. Or also, you don't forget, there are some more things that is called indels, insertions and deletions, copy number variations, and structural variants, which are present in only a small fraction of cells within an individual. And if you want to understand these five things, I have presented the figure which summarizes all these things. For example, whole genome sequencing, which is basically sensitivity traditionally zero point 0.1 to 1%, molecular barcoding greater than 0.001%. Then whole genome sequencing, 22,000 protein coding genes. These days, there are even more, almost double or more than double. And 1 to 2% of the genomes are targeted, that is coding sequences. Targeted sequencing, highly customizable, Guardant 360DX, companion test of ELA, analysis of ESR1 gene included. And this same thing can be done, for example, BRCA1 and 2 mutations, that is targeted sequencing. Evolution of the ARMS-PCR is also here, and you can go for comparative experimental work also. Now, what are the goals in general involved with NGS applications? Key opportunities and challenges are associated with clinically diagnostic genome sequencing. Application examples are also included here. Aspects of clinical utility, ethics, and consent are required. Analytic and post-analytic considerations are very important, and professional implications are also available. Now, cost of NGS, how it has decreased so far. Now, in 2025, the whole genome sequencing will cost also up to 500 to 700 US dollar, which was earlier in 2015, 5,000 US dollar. Now it has reduced almost 10 times lower. So NGS platforms are diverse platforms, differ in design and chemistry, fundamentally related sequencing of thousands to millions of clonally amplified molecules in a massively parallel manner. Orders of the magnitude more information will continue to evolve and has already been evolved. Attractive for clinical applications, individual or personalized sequencing assays, costly and laborious serial gene by gene analysis are possible. So there are a number of companies these days which are offering these NGS services at large scale, and also to the researchers in academy as well as in the industrial zones. Now, what are the basically examples of NGS? So, for example, here, discovery tools, single cell gene disorders, and causative mutations for multigenic diseases. diagnostic advancement for the diseases with overlapping symptoms. Multiple possible syndromes are also possible. Inherited conditions are challenges, and inherited conditions include challenges and opportunities. And you can understand here in monogenic disorders, novel missense mutations, germline mosaicism, the structural aberrations, imprinting effects, epigenetic factors, and multifactorial diseases like risk loci more often in non-coding or intergene regions, pathogenicity of variants often unclear, less testing versus monogenic disease. Reference human genome cataloging of variants are more test offerings are required. Now, what are the most notable applications? For example, cancer susceptibility genes prediction or possible risk assessment and management. Somatic and driver mutations are easily predicted. MicroRNAs are also predicted. For somatic and driver mutations, you can look for a number of research papers which were published in the New England Journal of Medicines and also in Nature Genetics, which have clearly given the fundamental basis for predicting the somatic and driver mutations. Then you can look for the epigenetic changes like transcription factor binding site methylations, alterations in gene expression could be calculated. Molecular profiling, tumor subtyping is possible with the use of NGS. Now, patient stratification could also be done. Predictions of therapeutic response for personalized treatment are easily predicted. Therapeutic monitoring is quite possible and easily implementable. So, prognosis is one of the major applications these days for NGS. Now, NGS application examples here, you can see some other things. Mutation panel screening could be done. Exome and transcriptome screenings are easily done with short time period. Genome sequencing comparison to normal tissues and reference samples could be done. Now, the analysis part is basically quantitative nature of NGS, like improvement versus chip technology, gene expression tests, and germline DNA characterization and somatic changes. Transcriptome and methylation profilings are easily done by using a single and rapid and cost-effective platforms. NGS application examples are in terms of other consideration or perspectives. Different NGS platforms have different capabilities. RNA and DNA sequence changes, DNA copy number changes, variations, DNA rearrangement, RNA expression profiling or transcriptome profilings, and then methylations are easily carried out and frequently carried out. A single method usually provides only part of this variety of information. Cost, specimen type, and application considerations are very important. These days, the most common application for NGS are single cell sequencing. Now, the NGS application examples in here, significant false positive rates are present. Mutation confirmation usually by Sanger sequencing will platform evolution eliminate it. So you need to go for cross validation if you are not sure. Variable percent of tumor cells and variable percent of tumor cells with presumably secondary mutation may overlap with NGS false positive rate. So numerous heterogeneous aberrations, for example, oncologic applications need algorithmic development, and now there are a number of algorithms already available. So, clinical utility, balance of net health benefits versus harm, NGS transformative for personalized treatment of disease, clinical indication includes test rationale, patient population, and clinical scenarios. Principles of comparative effectiveness requires individual or evidence-based approach for each patient. Clinical utilities and challenges here, it includes which variants are clinically actionable, development of evidence-based scientific standards to evaluate the utility in different patient populations for accurate risk estimation and prediction, risk of over-interpretation, unnecessary medical action, unwarranted psychological stress, careful selection of patients for genome sequencing and genetic counseling are crucial. So informed consents and ethical considerations are mandatory when you are working for human sample or animal models. So you need to create the patient awareness of the benefits and the harms. No specific guidance exists so far. Potential for anxiety and uncertainty exists, especially for variants of unknown significance. Discovery of incidental findings unrelated to the disease are in question. Now, there are also some regulations, assay validation, and reference materials. FDA lab developed tests, validation, lab approved cleared tests for verification. Then there are more things, so considerable uncertainty regarding regulatory pathways for NGS tests. Now, the challenges sequence are not truly complete, gaps in reads, GC rich regions, so bioinformatics limitations with indel variant callings persist. Gold standard comparisons are available, so you need to go for some old experimental approaches which can easily benchmark the prediction from the NGS. Regardless, all the NGS steps must be evaluated and quality control metrics must be in place. Is a sequencing portions of reference genome sufficient, or you need to think some different? Development of reference materials for the meaningful validation is the key to success for NGS application. Now, there are a number of organizations which you can also check. For example, CDC, then CLSI, ACMG, CAP, and AMP. What are the bioinformatics applications, tools, and techniques? There are hundreds of tools and approaches. 90% of the tools are freely accessible, but there are some commercial tools also. Depending on your lab budget, you can decide which tools or modules you are going to implement for your bioinformatics analysis. So evaluation of variant positions called involves queries for all the known relevant databases. Lack of databases curated to accept clinical standards likely the most significant challenge in managing and reporting genome sequencing data. EHR considerations, test ordering, archiving of NGS reports, patient consent, and data reinterpretations. So, post-analytical considerations, expert interpretation and guidance correlation of age, gender, clinical presentation, and family histories. Team approach ideal, like pathologist, geneticist, and other providers are required. Proficiency testing and alternative assessments are challenging and required. Proficiency testing schemes based on NGS methods versus specific genes are likely. So genomic education, the goal is to provide trainees with solid grasp of current concepts with broad range of opportunities. AMP, CAP, ACMG, and others working in this area. Training residents in genomics, curriculum designed to be adopted by any pathology residency. Trainings needed outside the fields of pathology and genetics is needed. So now what I need to summarize, that you have to understand the basics of NGS application for genomics understanding and quite broad range of precise and high accuracy application and predictions. So you need to understand the fundamentals of NGS application and what will be the project goal and which tools you need to select depending on what is your final goal. So this is overall what you need to understand the fundamentals of NGS and its application. Now I will be continuing the details of the different types of NGS application and methodologies and how to implement them easily without buying any commercial softwares. So thank you very much.